Slides of the dual porous media, quite clean as it went on. What we're seeing out of these systems is what looks like compost tea. And it can be quite dark to start with, and then it gets to be sort of lighter brown. And that is likely, although we're just fully characterizing that now, looking into it, but it's organic, dissolved organic material. Humic acids, fluic acids, and this can be very important because copper can be, as I mentioned, copper can be quite toxic at very low levels in salmon. Well, there's some research and some work that's been done previously, and some of the experiments we're doing now indicate that dissolved organic carbon might be very important for locking up the copper so that it is not available. It's not toxic. It's not available to the fish. So that's something that we're really still trying to understand, but might very likely be a very important difference between conventional stormwater management facilities and bioretention, is this dissolved organic carbon piece. And you can see, it increases quite a bit as the water moves through that soil column. And that's not surprising. I mean, it's soil. It's a media. And there's that organic component to it. So this is actually the second storm. Last year in November, we ran our first experiments where we ran storms through it. What we discovered was, just to jump ahead a little bit, was that our stormwater is quite clean. Bummer. We wanted really dirty stormwater. So we're in the process of figuring out how to make really dirty stormwater artificially. So we're doing that right now. But these were the second storm we ran through our system when and then we discovered again that the water was quite clean. But again, a good test. And probably, this is probably stormwater that's not that uncommon out there. It's clean. So it was a good look. Again, it's kind of this leaching question. And down below here are the mean and median of the effluent for each of these columns. And these are just the 60-40, because this time, all the money we had was just to test these 60-40, 60% sand, 40% compost. And once again, we look across this. Here's the influent monitoring station. This is the quality of the water going in to the 60-40 treatment. And we can see again, here's quite low, and we jump up about an order of magnitude in phosphorus. So we're exporting phosphorus out of these systems, out of this particular treatment. And so we have work to do there. The total phosphorus also jumping up quite a bit. And TSS, the total suspended solids, you can see in here, holding somewhat constant. No dramatic difference there. Again, so this is our nutrient end of it, and then metals. And here's where where we really have a sort of level of concern on this material. In influent monitoring station, again, you can see, we'll focus here on dissolved copper. This is where we focus a lot in this region. The dissolved copper is what can be particularly toxic. The particulate, maybe not so much, because it's, again, bound up in that particular form. And so here's our influent monitoring station again. And we're, quite clean. This is parts per billion. And you can see each of the systems leaching some copper. And again, we're still down fairly low levels, almost parts per billion. And so how well can we do with this as we try to improve the performance? It's yet to be seen. And I'm not sure, because we are at very low levels. Zinc, wow, zinc's a very good story. Again, here's what we had coming in to our system, and then some of these are actually let's focus on the dissolve that's detect, below detect level actually. So stellar performance on zinc. So this is our first storm, first clean storm. We're on the process now of ramping up to phase 2, which is creating our own synthetic stormwater that's dirty. So I want to just pause here for a second and mention how important this is for the valuation that we're talking about. If we don't understand the performance of these systems, it's going to be very difficult to get to correctly value and understand the cost and benefit of these systems as we move forward. On the positive side, bioretention systems are systems that are designed to manage a broad suite of contaminants. Big difference between a sand filter that's just phosphorus. All you're trying to manage is phosphorus. Very narrow set of constituents. These systems, apparently, for hydrocarbons, and we're just starting on getting our first data back on this, but for hydrocarbons, some metals, bacteria, we don't know much at all about organics. But for a fairly broad suite of constituents, these systems appear to work beautifully. But there appears to be three really challenging things. And again, it's the nutrient and copper question. And that's, if we can get to a point where we're going to optimize these systems and really get to that nitrogen, phosphorus, and copper question, and retain all these positive characteristics of these systems now, we'll really have the ultimate filter. We will have the best filter known in stormwater management. That's very different because it's dealing with a whole suite of contaminants. That's very different than how we currently manage, where we're generally focusing on one or two or very narrow band of constituents. So in our evaluation, that has, I think, dramatic implications for how we value these systems. Let's say 10 years from now, all of a sudden we have bacteria regulations, or we have regulations pinpointing cadmium, or polycyclic aromatic hydrocarbons. And we haven't thought about this, dealing with those constituents at all. And we've installed lots of systems that deal with phosphorus, or it deals just with one thing. We're going to have to be thinking about a lot of retrofit, a lot of how to manage, how to deal with that, as opposed to trying to work on a system that has a much broader application across a wider spectrum of constituents. So I'm trying to bring it back to this valuation question. Important questions as we move forward and try to understand performance, correctly value, and particularly think about future implications. It's not, I think we're probably on the verge of bacteria guidelines in this state. Where we stand in other parts of the country. So it's really important to get at this performance question in the context of evaluation. Okay, so we're sort of done with the mesocosms for the moment. We also have a whole permeable pavement setup, again, full-scale replicated porous asphalt here and down below, just down in this area, pervious concrete. And so on the porous asphalt installation, we're really focused on water quality. We're measuring pollutant levels at 2 levels. Treatment after the water has just run through the pavement itself. We collect water there, and then water quality is as moved through the pavement and the aggregate base, the gravels are used. So we're really focused here on water quality treatment, and maybe this next week will be our first sample. So we're just getting started on that. On the pervious concrete, we have a number of different sampling scenarios going on there. We're looking at hydraulics, but we're also looking at water quality treatment as far as how, for example, metals. Is there a metal buildup in these systems over time? So these ports right here, for example, we can access the pavement itself, of course, but then we can burrow down into the aggregate and the subgrade and look at metal concentration over time. So we're just getting started on that. We have monitoring wells looking at groundwater. So this is all stuff that's just getting started. We don't really have much in the way to report on data. This is just a look at the facility. Here's the asphalt cells with the collection, with all the collection ports and the measurement devices and the pavement. So what we've done here is look at infiltration rates of the pavement concrete and asphalt. And you can, just sort of looking here across this, we're starting at very high rates. Again, here we're at centimeters per hour. Yay, but quite high. We're starting off with quite high infiltration rates on these systems. So we'll be measuring these over time and see how that degrades or doesn't degrade. And but this is as far as we've got with these systems right now. So I want to finish up with just a couple things, and one is what we're going to be doing, what we're working on now, and sort of the future research, most immediate future research. And this is biological effectiveness of green stormwater infrastructure. So understandably, jurisdictions and the federal government, the state governments, the local governments, they're concerned about their permit. How do we meet our permit requirements? How do we meet these effluent numeric standards? We're concerned about that and asking those questions, but what we're really interested in is, can we design and build stormwater management facilities that actually protect aquatic resources? Forget the numeric standards, because is there anybody here from EPA? Yes, Dina. I know this sounds, I don't mean this to be antagonistic. There is very little connection between our numeric standards and protecting our biological aquatic resources, the bioresources. And one reason is because we have very simple tests. It's not because there's bad people making those regulations. It's because we only have a certain amount of money, we only have a certain amount of time, and it's very difficult to do very in-depth studies on the effectiveness of actually protecting aquatic biota. So we've looked at, does this stuff kill the organism or do they live? That is extraordinarily coarse and blunt instrument for understanding how well are we doing as far as protecting aquatic resources. The sublethal effects, that is a degrading of the ability of that organism to sense its environment, to find food, to avoid prey, all that also kills aquatic organisms. And so that's the question that we're really starting to focus on. What is the actual biological effectiveness of these systems? So we're looking at, can we sufficiently reduce contaminant concentrations to both prevent lethal and sublethal effects? This is by far, in a way, the more complex question. Extraordinarily complex question. Step one, we're testing effectiveness using high throughput surrogates. So these are things like, instead of using salmon, for example, in test, we're using zebrafish. So that's all that means. It's a particular organism that is well adapted for lab studies. Step two, we're going to focus on salmon-specific direct and indirect impacts, both at the lab scale and then also moving out to the full-scale applications where we're, for example, hooking up synthetic stream channels to those mesocosm tanks. So small stream channels that have rock and gravel and rock and insects and fish in them, and running untreated and treated water through those synthetic stream channels. So I just want to sort of emphasize this point of sublethal effect and explain to you one of the here is, for example, some work on copper. And NOAA has done, this is not work we've done, or I've done, this is work that NOAA has done here at the Science Center in Seattle, and that we're working on now with them. But this is a level, micrograms per liter, levels of copper in the water. And this is, on the vertical axis here is this thing called percent freezing behavior. Salmon, when they are bit by a seal or a bull trout or something that, one of the predators, they emit a chemical. And that's an alarm chemical, and the surrounding fish will say, something wrong, and they freeze. They stop moving, because many of the predators are triggered visually. And so they'll stop moving, essentially lay dead. That's how the other fish avoid getting eaten. You expose salmon to low levels of copper, again, micrograms per liter. And you can see at quite low levels, the number of fish, the amount of time, how they freeze, how they respond to that chemical cue is dramatically degraded. And so these fish that are exposed to copper, when that chemical is released in the water in the tank with those fish, they just keep swimming. They don't sense that chemical. Those fish are highly likely to be predated as compared to their healthy, to the other healthy fish. So this is an example of the sublethal effect that we're working on, both with metals, hydrocarbons, and working to a large degree with polycyclic aromatic hydrocarbons now. It appears to be a particularly bad actor, are these polycyclics.
How does freezing become negative?
I'm sorry, question again?
You have the freezing behavior out there,
Yeah.
but the last measurement over there is
Yeah.
below zero.
This one down below zero here. This, I don't know why. This is Jen McIntyre's work, and I don't know why that's at zero. So these are extreme levels of copper for us. This is 20 micrograms per liter, and I don't know if that's an aberration of the graphing, but for us, this is the important piece right
I was
here.
just wondering how you measure the freezing behavior. I would say zero would be like no acidity.
No, that's right. No freezing. That's correct. So this would be, no response. So to some degree, some still, there may be a reduced amount of swimming. We get down to this point, and basically that organism will continue to swim, unaware of that particular chemical in the water. All right, so just an example of some of the sublethal effect and the work that we're doing on looking at that issue. So one of the things we're doing right now is looking at collecting stormwater and running that stormwater through columns that we have in the greenhouse. Some of these with plants, some of these without. They all have the same media. And that media is that 60% sand, 15% compost, 15% shredded bark, and 10% WTRs. It's one of the medias we had outside, and we've been measuring. That media is in these tanks. We are then using this, highway runoff and exposing organisms to that highway runoff, and then exposing untreated, and then exposing those organisms after it's been treated through. So I'll give you one short and pretty dramatic result from this. What we found so far is that the organisms, salmon, was exposed to the highway runoff, 100% mortality. All the fish that were exposed to that highway runoff died. 100% of the fish that were exposed to the water that went through the columns survived. And right now we're starting to look at the sublethal effects. So stay tuned. That's going to be I'm so excited to see that information, because I would expect there's some sublethal effects, but I don't know, we'll see. But the first experiment, quite dramatic, in that it's black and white as far as behavior, or as far as survivability. So we're going to be running a number of different things, looking at experiments, looking at this coho survival. This is what we're doing right now, as well as other organisms, and then other sublethal effects as well. So that's all going on right now. So just to finish up, next steps. Larger scale biological effectiveness studies. So this is where we're going to be hooking up those, hooking up synthetic stream channels to the mesocosms and ramping up in complexity and organisms, a number of organisms. Really interested in developing partnerships internationally, particularly, to expand research, particularly on the plant-soil interactions. This slide is, there we go, okay. These lines are all divided, and particularly rhizosphere studies. It appears that where a lot of activity is happening in these systems, as far as how well they perform, is in the rhizosphere. So that zone right around the roots. A lot of very biologically active, a lot of chemical interaction, and very important in how these systems behave. So we're really looking to expand that work and use these systems that are really being underutilized right now for looking at different plants and how the different soils evolve, how those plants are interacting with those soils over time. So that's a big one that we're looking at expanding into. And then novel amendments and polishing filters. So the novel amendments, again, things like biochar. Very interesting material. And biochar is virtually, essentially any organic material that's burned in a low oxygen environment at a particular temperature for a particular amount of time. Very interesting material, and it's also can be a waste product. And so we're starting experiments looking at that. But also, there's parts of bioretention that we have not paid much attention to, and that is when you have a drain, an underdrain, and you put that underdrain in some gravel or sand. We want to start working on how do we use that layer for a final filter. Perhaps we can polish out that last bit of copper or that last bit of phosphorus in that drainage layer. That right now, all we're using it for is put a pipe in. That's our focus. So looking at working on that. And then, I've already mentioned, I know we're a couple minutes over time, so I'm not going to talk any more about this. But again, I just want to emphasize that all this stuff, this performance and how these things behave, understanding that is critical for this valuation exercise. And I would also, again, ask in the valuation exercise that we can think about future implications of this and what we might have to manage in the future, and that the systems that we think about, that we consider them instead of 10 years from now having to do a bunch of retrofitting and catching up with new regulations. All right, that's it.
Since we are running late on time, are there any burning questions? I'm not trying to encourage you to ask questions, but if you have some. Thank you. And I'm guessing that you'd welcome visitors to the Stormwater facility, Curtis? Someone wanted to come
Absolutely.
visit you sometime?
Yeah. Tomorrow, would you like to come down tomorrow?
No, we'd love to. It's just the train doesn't stop. But we'll keep our eye. I'm sure everybody will keep their eyes on the research that's coming out of that. So I'd just like to conclude by thanking everyone, all the people who contributed, all the volunteers. I forgot to mention Lisa earlier on, all the organizers, Anja and Rena, funders, and for all of you for coming from where you come from, whether it's from across town or across the Atlantic. So we hope that this has been useful to you or build a good foundation for the rest of the tour for the days and for the rest of the continuation of application of marine stormwater infrastructure in the Pacific Northwest. So here's what's next. We have about 10 minutes